Warning: your DNA is 8% alien virus. By the time the fever hit, the damage was done. A virus slipped into an ancestor's germ cell and wrote itself into the family line. Today, about 8% of your DNA, more than triple the 2% that codes for proteins, is viral code wired into your cells. It built your placenta, helps wire your brain, and when it misfires, can light up diseases like multiple sclerosis and ALS. The wildest twist? Scientists can flip parts of it back on to make tumors look like infections and get the immune system to attack. To see how that happened, we need to rewind. Endogenous retroviruses are ancient infections that invaded sperm or egg cells, got copied into the genome, and now passed down like eye color. They're alien in origin, but deeply domestic in function. Regulators, switches, sometimes hazards. Here's where we're going. An infection becomes inheritance. Fossils become tools. Pregnancy borrows a viral fusogen. The genome builds firewalls. Cracks appear in disease, and medicine learns to weaponize viral echoes. Keep the tone grounded, curious, cautious, a little rattled. To understand the debt we owe these ghosts, meet the ancestor who accidentally adopted a virus as family. Patient 0, the night a virus became family. It starts small. A bite, a scratch, a shared fruit. An ancient primate catches a retrovirus, the kind that doesn't just hijack a cell, it rewrites it. Most infections end in a dead end. The body clears the virus or the host dies, and the code goes with them. But not this time. The virus carries simple instructions. Its RNA genome enters a cell, and using reverse transcriptase becomes DNA. Integrase threads that DNA into the host's chromosomes. Long terminal repeats, LTRs, stamp the start and end like bookends. In the middle sit the classic viral parts. GAG for the capsid shell, POL for enzymes including that reverse transcriptase and integrase, and ENVER for the protein that fuses membranes. It's a tidy kit for breaking and entering. Here's the 1 in a million break. The infection lands in a germ cell. Not a skin cell, not a liver cell, an egg or a sperm. That's the only doorway to forever. When that germ cell becomes a zygote, the integrated provirus hitches a ride into every new cell as the embryo divides. Suddenly, a one-night stand is a lineage. Within a mating season, that family carries a quiet passenger. Some insertions vanish by chance, others ride a wave of reproduction. If that retroviral scar doesn't kill, it drifts. If it helps, by changing when a nearby gene turns on, or by blocking entry to cousin viruses, it might even spread faster. Over generations, neutral mutations hit the GAG, POL, and reading frames. Frame shifts, stop codons. The once infectious provirus decays into a fossil. But the LTRs, those regulatory bookends, can still talk to the host machinery. The genome keeps receipts. At each insertion site, you can see target site duplications, little footprints flanking the provirus. Some families of these elements prefer certain chromatin states, slipping into open, actively transcribed regions. Others pepper heterochromatin, like confetti in the silent parts of the genome. Either way, the insertion creates a new regulatory possibility. A promoter where none existed, an enhancer with a fresh transcription factor binding site, or a decoy that soaks up repressors. Meanwhile, there's a counterattack brewing. Host cells learn to spot viral hallmarks and clamp down. KRAB zinc finger proteins evolve to recognize specific sequences in these newcomers. They recruit TRIM28, also called CAP1, which calls in histone methyltransferases to lay down H3K9Me3, heavy repressive marks. DNA methylation spreads like wet cement, hardening the silence. In germ cells and early embryos, small RNA pathways, pRNAs, guide machinery to matching sequences and warn, shut this down. ApoBec enzymes mutate cytidines into uracils on single-stranded DNA during reverse transcription, leaving pockmarks in any latecomers. The firewall rises. But firewalls with doors are useful. In the right tissue, an LTR can be a well-placed access point. Some LTRs carry binding sites for STAT1, the switch that flips when interferon floods a cell during an infection. One viral remnant family, MER41, seeded interferon-responsive enhancers near genes like AIM2, APOL1, IFI6, and SETIM1. What began as a threat became part of the alarm system. The host didn't just defend, it repurposed. Picture a troop of primates after that first season. An infant tugs its mother's fur. Inside its cells, the retroviral scar sits in the same chromosome region the mother carries, the same region the father lacks. In a few generations, by drift and chance, the insertion becomes common. If it's harmless, it may hang around as background noise. If it subtly boosts resistance to a circulating virus by interfering with its receptor, or if it reshapes expression of a nearby gene in a way that improves survival, it can fix in the population. Everybody has it. Now, it's not an infection, it's a locus in the species. Time blurs the line between self and invader. Recombination between the LTRs can pop out the interior viral genes, leaving a single solo LTR behind like a signpost. The two LTRs at the ends also slowly diverge from each other, ticking away like a clock that lets geneticists estimate the age of the insertion. Younger families, like HERV-K, still show relatively fresh sequences, and in some cases, residual activity. Older ones, like HERV-H, are nearly all fossils, but still lend regulatory phrases to the genome's grammar. There's a moment when this isn't just molecular, it's personal. The ancestor with the fever mates. A zygote forms. One cell becomes 2, then 4, then a blastocyst. The provirus copies with every division, cemented into the origin story of a person who will never know it's there. That scar, in one lineage, will one day regulate an immune gene during a winter outbreak. In another, a descendant's brain will open a set of LTR-driven enhancers at just the right developmental hour to sculpt a cortex subtly different from a cousin species. A threat became an option. Of course, not every passenger is a gift. Some insertions slice into the middle of important genes and get purged by selection. Others sit close enough to matter, but far enough to survive, waiting on the epigenetic leash. The host and the remnants learn a tense coexistence. Most sequences locked down, a minority kept poised ready for regulated use. This was the night a virus became family. Not by destiny, by mechanics, chance, and selection. What nobody knew in that forest was how far the consequences would reach. Because one of the greatest human upgrades would come from the most viral of tricks, fusing cells. And that story starts where every one of us started, at the placenta. Viral tools, human upgrades, exaptation in action. To understand the upgrade, remember the kit that virus left behind. LTR bookends with promoter power, and a cargo hold that used to make infection possible. Once the replicating parts broke, what remained was a pile of switches and sequences. Evolution doesn't waste a good switch. Here's the stakes check. If a viral remnant helps survival even a little, shaving a day off an immune response, nudging a developmental gene at the right hour, it's more likely to stick around. If it's harmful, layers of silencing pile on until it's effectively mute. Most are quiet fossils, but a fraction got hired. Start with the alarm system. During infections, interferon floods a cell and flips STAT1 and other transcription factors. Some ERV LTRs already carry binding sites for those factors, because the original virus needed to turn on fast when a cell's machinery was active. The host stole that bias. One ERV family, MER41, landed near immune genes and now acts like a row of fire alarms. When interferon spikes, MER41 enhancers help light up genes such as AIM2, APOL1, IFI6, and SECTUM1. What used to be a viral on-ramp became part of a human early warning network. Meanwhile, the brain found its own uses. Development is all about timing and place. Turn this gene on in this cell type for a narrow window, then shut it down and hand off to the next program. LTRs are natural candidates for alternative start sites and enhancers. In the developing cortex and hippocampus, clusters of ERV-derived switches open in waves, recruiting transcription factors that weren't even a concept when the virus first jumped in. The result is subtle. A tweak in neuron subtype ratios, a change in dendrite branching thresholds, differences that add up across many loci to tune learning, attention, and memory. It's not one dramatic lever, it's 1,000 sliders. Mechanistically, the adoption is hands-on. A dormant LTR sits in heterochromatin, silenced by H3K9, Me3, and DNA methylation. In a specific tissue, pioneer factors bind a motif within that LTR, recruit coactivators, and repel repressors. Nucleosomes move. The LTR becomes an enhancer looped to a nearby promoter through cohesin and CTCF, changing 3D chromatin architecture. Or the LTR's core promoter is repurposed as an alternative transcription start site, producing a longer or shorter first exon that alters translation efficiency or protein localization. Sometimes the LTR launches a long, non-coding RNA that scaffolds regulators to a neighborhood of genes, modulating an entire program. Get this, the immune system didn't just adopt the alarms, it borrowed viral tricks for blocking. Some ERV and remnants, though crippled, kept receptor-binding fragments. Expressed in certain tissues, they can act as decoys that occupy viral entry receptors, lowering susceptibility to related pathogens. It's a simple move. Jam the lock with a broken key. Across mammals, independent captures of NVA-like sequences keep showing up, hinting at a strong selective payoff. The payoff isn't only protection. Tissues under heavy regulatory demand, placenta, brain, immune cells, early embryos, show enriched ERV-derived regulatory activity. It's like evolution-sprinkled accelerators, where rapid, context-dependent tuning matters. Humans and chimps share many ERV fossils, but the same family can be wired differently in each species. Different LTRs opened, different factors recruited, different loops formed. Over millions of years, that rewiring contributes to lineage-specific traits. You can see it in stem cells, where certain HERV-H elements mark active pluripotency networks in humans, more than in other primates, shaping how early embryos set up developmental competence. There's another layer, responsiveness, because these sequences evolved to be sensitive to cellular states, stress, inflammation, hormones. They serve as built-in context sensors. Under interferon, a subset of LTRs wake to boost antiviral genes. Under neuronal activity, activity-dependent factors bind neuron-specific LTRs to fine-tune synaptic genes. Under hypoxia, a different subset may engage. The host didn't invent thousands of new switches from scratch. It sifted through the viral junkyard for parts already wired to respond. But there's always a but. Reusing hacker tools means living with hacker risk. The same LTR that boosts an immune gene in a macrophage could, if misexpressed in a neuron or a germ cell, create noise or toxicity. So the system evolved layers of breaks. KRAB-ZFPs that recognize particular LTR subfamilies, TRIM28 to enforce repression until the right factors arrive. PRNAs in germ cells to keep the heritable line clean. It's a negotiated truce where the host sets strict curfews and occasional work permits. And here's where it gets dramatic. Among all the exapted tools, one set stands out for changing mammalian life. Captured M genes that fuse cells. Viruses use MV to merge with host membranes. Mammals repurpose that fusogenic power to build a new tissue, the syncytia trophoblast of the placenta. In humans, two of these MV-derived genes took center stage with different roles and timing. We call them Syncytin 1 and Syncytin 2. Those names sound technical, but their effect is not. Without them, placental fusion fails, nutrient and gas exchange stalls, and live birth as we know it collapses. A viral battering ram became the architect of early life. We'll zoom into that fusion engine next. The placenta's secret, syncytins and live birth upgrades. Before there was a heartbeat, there was a merger. A tiny ball of cells reached the uterine wall and needed a way to plug in, share oxygen, share nutrients, and avoid getting mistaken for an invader. That plug isn't a simple connection. It's a living interface called the syncytia trophoblast, a continuous, multinucleated sheet that fuses from many cells into one barrier. And the engine that does the fusing came from a virus. Viruses use invric proteins to fuse their membranes with host cells, pry open a route in, and dump their genetic cargo. Mammalian placentas hijack that exact talent. In humans, two invade-derived genes stepped into essential roles with different timing and duties. Syncytin 1 from the ERV-W1 family and Syncytin 2 from ERV-FRD1. They aren't backups. They're co-leads in a choreography where a single misstep can collapse a pregnancy. Here's how it plays out. Trophoblast cells at the outer layer of the embryo differentiate into cytotrophoblasts that line up against the maternal tissue. Syncytin 1 is expressed on their surfaces. When its receptor aligns, like two halves of a zipper, the protein snaps into an active shape, brings membranes within kissing distance, and drives them to merge. Two cells become one, then another adds on, and another. The sheet grows into a syncytium, the fused skin of the placenta, studded with microvilli that increase surface area for exchange. A viral battering ram became a construction crane. Syncytin 2 runs a parallel job with quieter but critical effects. It also carries fusogenic power, but its expression pattern and regulatory control differ, and its protein includes an immunosuppressive domain, an ancient viral trick turned guardian. At the maternal-fetal interface, that domain tones down local immune activation, helping the mother's immune cells stand down in a place that, frankly, looks like a graft. Together, the two NV-derived proteins balance force and tolerance. Build the barrier, then keep the peace. The stakes couldn't be higher. If fusion fails or the immune truce cracks, conditions like preeclampsia, fetal growth restriction, or early loss can follow. Researchers have found altered levels of Syncytin 1 or Syncytin 2 transcripts and aberrant methylation around their LTR promoters in complicated pregnancies. It's a reminder that the genome's locks and keys aren't philosophical, they're medical. Now widen the lens. This wasn't a one-off miracle unique to primates. Across mammals, evolution kept stumbling onto the same hack. Rodents captured one NV gene for fusion, then another later. Ruminants like cows did it independently, bats did it, carnivores did it, different viruses, different times, same solution. Grab INV, domesticate it, and build a placenta that fuses. That convergence screams utility. When nature finds a working part in the junkyard, it keeps pulling the same part. This convergence hides a second lesson. There's more than one way to tune the same organ. Lineages that domesticated different NV genes show differences in placental structure and depth of invasion. Shallowly invasive placentas, like in some ungulates, solve nutrient transfer with surface area and vascular architecture. Deeply invasive primate placentas lean on intense remodeling of maternal arteries. The shared language is fusion. The dialects are species-specific. Of course, viral tools bring viral liabilities. The same fusogenic power that helps build tissue can get misapplied. In several cancers, melanoma, breast, germ cell tumors, HERV-derived NV proteins, including HERV-K and members of the HERV-W family, show up out of context. Cell fusion in tumors can create hybrid cells with new traits. More motile, more drug-resistant, more aggressive. Some M proteins can also act as ligands for innate immune receptors, like TLR4, kicking off inflammatory cascades that reshape the tumor microenvironment. What built the nursery can also help an invader break walls. So why doesn't the placenta fuse everything in sight? Locks, and lots of them. The LTRs flanking Syncytin 1 and Syncytin 2 carry promoter sequences that respond to placenta-specific transcription factors. And those same regions are kept repressed in most other tissues by DNA methylation and repressive histone marks. KRAB-ZFPs patrol for the wrong context. TRIM28 enforces silence unless the right factors arrive. Even within the placenta, expression is spatially patterned. Cytotrophoblasts express, syncytiotrophoblasts maintain, so fusion proceeds where and when it's safe. There's a micro-story hidden in every pregnancy test. A few days after conception, as HGG rises, the embryo sends molecular messages to preserve the uterine lining. Meanwhile, on the cell surface, Syncytin 1 molecules are quietly zipping membranes together, turning hundreds of individual trophoblasts into a single working organ that will filter a mother's blood for months. No fanfare, no blueprint written by mammals from scratch. Just an old viral skill repurposed perfectly. One detail would matter later. These NV-derived proteins still look viral enough to the immune system to be recognized when they appear in the wrong place. That's part of why they can become red flags in tumors, and part of how medicine is starting to use them. But before we turn the immune system loose, keep one idea in mind. If a virus helped build the interface that kept you alive before birth, what else did viral code help wire after? The next clues aren't in the womb, they're in the brain. The firewall, how cells muffle the viral choir. We've been celebrating the upgrades, but upgrades come with guardrails. 8% viral code is powerful hardware. If it misfires in the wrong tissue, at the wrong age, with the wrong trigger, it can jam gene programs, fuse cells that should stay separate, or trip immune alarms. So cells built a layered firewall, fast, redundant, and paranoid. Start at the DNA. The simplest lock is a chemical tag, methyl groups glued onto cytosines, especially at CPG islands inside LTR promoters. Methylation doesn't change the letters, but it blocks transcription factors and recruits proteins that keep chromatin tight. It's the universal do not enter tape, renewed every cell division. Layered on top are histone marks. The key repressive badge here is H3K9, Me3, a trimethyl flag planted on histone H3 that tells packing proteins to squeeze that region into heterochromatin. Closed chromatin means the polymerase can't get in. The viral promoter can't shout. Who lays the flags? A specialized police force. KRAB-Zinc finger proteins, KRAB-ZFPs, are an enormous family of DNA-binding sentinels that evolved in part to recognize specific transposons and ERV sequences. Each KRAB-ZFP is a custom key that fits a handful of sequences. When it binds, it recruits TRIM28, also called COP1, which acts like a hub. TRIM28 pulls in SETB1, the histone methyltransferase that writes H3K9 Me3, and it scaffolds other repressors and chromatin remodelers. In early embryos and stem cells, TRIM28 sits like a blanket over young retroelements. Knock it down in mice, and endogenous retroviruses lurch awake innate immunity sparks, and microglia in the brain flip into an activated state. That's not a theory, that's what labs see when TRIM28 is deleted during development. ERV transcripts rise, ERV-derived proteins appear, and inflammatory pathways light up. Germ cells get a second firewall, small RNA patrols. The PEER-ANA pathway, built around Pee-wee proteins, uses tiny sequence guides to find transcripts from transposable elements and ERVs and slice them up. It's ruthless because any slip here becomes heritable. ERVs also direct DNA methylation machinery back to matching loci, reinforcing the lock at the source. In somatic tissues, other small RNA systems and RNA interference catch strays, while RNase H chews on RNA-DNA hybrids that form during errant reverse transcription. There's also a sabotage unit that scars incoming invaders. ApoBec enzymes, cytidine deaminases, rip Cs into us on single-stranded viral DNA during reverse transcription, causing hypermutation. Those edits cripple latecomer retroviruses and leave pockmarks even on ancient remnants that attempt to reactivate. If you read an ERV locus and see an unusual pattern of mutations, chances are ApoBec took a swing at it long ago. None of these locks acts alone. The genius is in layering. Most ERV copies sit under multiple forms of repression: DNA methylation, H3K9, Me3, KRAB-ZFP TRIM28 complexes, small RNAs. So if one mark erodes, the others hold. But not all marks are equal everywhere. Early embryos loosen global methylation, then reseal it with help from TRIM28 and SETB1. Placental cells deliberately open select LTRs. Immune cells hold certain interferon-responsive LTRs, poised, repressed, but close to ready, so a pathogen signal can safely crack the door. The catch is that lock's weather. Aging erodes methylation fidelity, the pattern phrase. Chronic stress and inflammatory cytokines can tug at chromatin. Interferons, the same molecules that help fight viruses, can also flip on ERV-linked enhancers like MER41 to boost antiviral genes. That's by design in macrophages, less so in neurons. In diseases where global methylation is reduced, like many cancers, swaths of ERV loci unsilence together. Double-stranded RNAs from antisense sense ERV transcription pile up. Sensors like MDA5, TLR3, and the She-gas sting pathway smell virus and trigger interferon programs. In tumors, that can be a feature. We'll use it to make cancers visible. In healthy brain tissue, it can become a fuse for neuroinflammation. And the brain firewall is delicate. In mouse models, when TRIM28 is deleted in neurons or during development, ERV activation correlates with microglial activation and expression of ERV-derived proteins in adult brains. The innate immune response doesn't care that these sequences have local citizenship. To sensors, viral nucleic acids are viral nucleic acids. That tension, citizen by birth, suspect by pattern, defines the entire relationship. Context matters. In the placenta, Syncytin 1 and Syncytin 2 LTRs are demethylated and opened by trophoblast transcription factors. In nearly every other tissue, those same promoters are locked down. KRAB-ZFP repertoires differ between species and even individuals, shaping which ERV families stay quiet and which can be coaxed for regulation. That diversity can be protective, more keys for more doors, but it also means some people's firewalls might be leakier at specific loci, changing risk profiles for autoimmunity, infertility, or cancer. Think of it as a choir most of us never hear. Thousands of voices, trained to stay silent, with a few soloists licensed to sing in carefully chosen rooms. The conductor is epigenetics. The ushers are KRAB-ZFPs and TRIM28. The soundproofing is methylation and H3K9 Me3, and the bouncers are Pirnas and Apobec. Most nights, the hall is quiet and the show runs on time, but a power surge, aging, infection, chemotherapy, stress, can pop a circuit. A handful of mics switch on. Sometimes that noise becomes a life-saving alarm, sometimes it becomes harmful feedback. We'll see both. First, the harm, when ghosts wake and push biology out of tune. Then, the gambit, using that very noise to flag tumors for destruction and disease. While the firewall usually holds, there are moments it slips, and that's when the ghosts start talking. You see it at the bedside, not just in sequencing data. A patient's first numb patch that creeps across a leg, a hand that won't stop trembling, words that get stuck mid-sentence. Doctors scan, biopsy, stain. Beneath symptoms, you often find a pattern. Epigenetic marks frayed, viral remnants awake, and an immune system reacting to shapes it thinks it remembers. Start with multiple sclerosis. In MS lesions, the inflamed scars peppering white matter, researchers repeatedly detect transcripts and proteins from a human endogenous retrovirus in the HERV-W family. One particular N protein, sometimes called MSRV-NV, shows up on microglia and astrocytes in diseased tissue. NV is a viral envelope protein designed to engage receptors and change cell behavior. In the brain, this NV can bind innate immune sensors, including TLR4, and drive nitric oxide synthase and cytokine release. That pushes microglia toward a pro-inflammatory state, amplifying the damage. Patients don't feel a viral protein on glia. They feel fatigue that flattens a day, heat sensitivity that erases function, and relapses that leave dents. Now ALS. In motor cortex and spinal cord neurons from some people with ALS, HERV-K, one of the younger, more intact families, shows evidence of transcription and even INV expression. Neurons are terrible places for viral echoes. They don't regenerate easily. And their neighbors, microglia and astrocytes, respond to ERV-derived nucleic acids and proteins like a siren. The result is a local storm. Interferon up, complement pathways on edge, synapses pruned where they shouldn't be. It's not the only path to ALS, and it's not universal, but in subsets, the ERV signal isn't a rumor, it's a finding. How do these dormant fragments cause trouble? There are several routes. One is direct protein action, M fragments engaging receptors, acting like superantigens that bypass normal checks and yank immune cells into overdrive. Another is nucleic acid sensing. When ERV loci wake, they often produce overlapping sense and antisense RNAs that anneal into double-stranded RNA. To innate sensors like MDA5 and TLR3, dsRNA screams, virus. Some ERVs may even yield cDNA through residual reverse transcription, letting C-gas Ting pick up cytosolic DNA and pour fuel on interferon signaling. You get an antiviral program without a virus, a body fighting smoke from its own archives. Add crosstalk. In diseases and aging, global hypomethylation loosens more than one family of repeats. Line 1 element STIR2. Their reverse transcriptase can amplify the chaos, creating RNA-DNA hybrids and extracytosolic DNA that escalate sensing. Meanwhile, transcription factors unleashed by inflammation bind motifs in specific LTRs, like the interferon-responsive MER41 family, boosting nearby immune genes. That's helpful in macrophages. In oligodendrocytes trying to remyelinate, it can be derailing. Here's the uncomfortable part. Association isn't causation. ERV transcripts rise in inflamed tissue. Inflamed tissue is full of broken regulation. Are ERVs passengers, or are they pressing the gas? The answer varies by locus, tissue, and person. But the line from interesting correlation to actionable target is starting to fill in. In MS, a monoclonal antibody that binds HERV-W NV has gone through early trials. The logic is simple. If NV protein fans inflammation, neutralize it. Early phase results suggest acceptable safety and signals on brain inflammation markers, though larger studies must test real clinical benefit. That's not proof that a virus causes MS. It's a test of whether dialing down a viral echo cools the fire. Cancer tells a parallel story with a twist. Tumors often carry global hypomethylation and chromatin disruption. Whole neighborhoods of ERVs unsilenced together, producing dsRNA and setting off intracellular alarms. That can be bad. Chronic interferon can select for resistant clones and drive exhaustion, but it's also a weakness. Those viral-like RNAs and peptides can make cancer look foreign. Some HERV-derived peptides show up on MHC molecules as neoantigen-like flags, unique to tumors. Immune cells can learn to see them. The same duality appears in pregnancy complications. The placenta runs on viral tools. Misregulate them, and you get risk. In preeclampsia, altered methylation at LTR promoters near Syncytin 1 correlates with reduced expression, impaired fusion, and heightened local inflammation. It's not that ERVs cause preeclampsia in a singular way, it's that the viral hardware requires precise control. Drift from that control has consequences. The brain's version of this drift is especially sensitive. After traumatic brain injury in humans, single-cell transcriptomics show glial populations flipping on ERV and other transposable element transcripts alongside innate immune genes. It's as if injury unmasked a second injury. The genome's emergency switches clicking wildly. In mice, delete TRIM28 during development, and adult brains show ERV-derived proteins and microglial activation. That doesn't map one-to-one onto human disease, but it nails the mechanism. Leucin, TRIM, 28s, COP1, and viral ghost wake. Wake ghosts and innate immunity answers. So what does a clinician do with ghosts? You look for leverage points. If an M protein is present and immunogenic, build an antibody. If dsRNA sits at the heart of a tumor's alarm system, force more of it with demethylating agents, then remove the brakes with checkpoint inhibitors. If an autoimmune flare tracks with interferon tone and ERV expression, test whether targeting the viral component reduces collateral damage. And if a patient asks whether stress turns on viruses, you translate biology into advice. Stress hormones and inflammation nudge epigenetic locks. Protecting sleep, moving your body, and treating infections promptly keeps locks sturdier. We're not erasing the 8%. We're learning where it leaks, when to plug it, and when to let it scream so the body hears the threat. Because the next move uses that scream on purpose. Turning the enemy on tumors, viral mimicry. Here's the pivot. Everything you've heard about ghosts waking and sparking alarms. What if we trigger that on purpose inside a tumor? Not in healthy tissue, in the cancer where epigenetic locks are already frayed. That's the idea behind viral mimicry. Make a tumor sound like a raging viral infection, so the immune system can't ignore it. Tumors hide. They downshift antigen presentation, recruit suppressive cells, and bathe themselves in signals that tell T cells to stand down. But most cancers also carry a tell: global hypomethylation and chromatin chaos. When methylation fades across the genome, ERV loci unsilence in clusters. They produce overlapping sense and antisense RNAs that fold into double-stranded RNA. To cellular sensors like MDA5 and TLR3, dsRNA screams virus. Some ERV transcripts can even lead to cDNA fragments that activate cGAS, STING. That's a native tripwire we can wiretap. Clinicians reach for drugs that erode methylation further, but strategically. Low-dose DNA methyltransferase inhibitors, like azacitidine or decitabine, don't just hit oncogenes. They lift repression across swaths of ERV sequences in tumor cells. Within days, dsRNA accumulates. Interferon pathways switch on inside the cancer, upregulating antigen presentation machinery and cytokines that call in immune cells. The tumor, which spent years turning down the volume, suddenly blares a siren. On its own, that siren helps, but the real punch lands when you pair it with checkpoint inhibitors, antibodies that block PD-1, PD-L1, or CTLA-4, the brakes tumors use to exhaust T cells. It's a 1-2, make the tumor look infected, then release the restraints on T cells so they can respond. In some melanoma, ovarian, and colorectal cohorts, higher ERV expression inside tumors tracks with better responses to checkpoint therapy. It's not magic, it's visibility. T cells attack what they can see and what looks dangerous. There's another advantage. ERV-derived peptides can be presented on MHC like neoantigens, but with a twist. Many are absent or at vanishing levels in normal tissues, yet abundant in specific cancers. That makes them tumor-selective flags. If you can identify a set of HERV-K derived or HERV-H derived peptides reliably presented in a cancer type, you can train T cells against them or build vaccines that prime the response. Early efforts are mapping these ERV immunopeptidomes, catalogs of what fragments appear on tumor cell surfaces, so therapies can be aimed without spraying the whole system. This isn't without risk. Interferon isn't a toy. Push it too hard and you can create an exhausted T cell pool or collateral inflammation that damages healthy tissue. Not all ERV expression is equal either. Some tumors raise ERV RNAs but fail to process and present peptides efficiently. Others trip alarms but quickly adapt, upregulating new suppressive pathways. That's why timing and dosing matter. Short pulses of demethylating agents to spark dsRNA, followed by checkpoint therapy to capitalize on the window, may work better than brute force. Patients aren't lab graphs. Take a woman with recurrent ovarian cancer. Standard chemo buys months, not years. In a trial, she gets low-dose azacitidine to stoke ERV dsRNA, plus a PD-1 inhibitor. Her tumor biopsy a week in shows interferon-stimulated genes up, MHC class 1 for stored on cell surfaces, and a mosaic of HERV-derived transcripts. 2 months later, scans show shrinkage. It's not every patient, but in responders, ERV expression isn't just background noise, it's part of the signature of vulnerability. Stratification becomes key. If you can profile a tumor's ERV landscape before treatment, how many loci are active, which families, what dsRNA burden, you can predict whether viral. Mimicry will light a fire or just smoke. Single-cell RNA-seq helps separate cancer cells from stroma. ATAC-seq reveals which LTRs are open, and immunopeptidomics confirms presentation. The goal is a simple clinical readout. High ERV score, likely responder. Low score, try a different lane. There's movement beyond small molecules. Researchers are exploring oncolytic viruses engineered to amplify ERV expression in tumors or deliver INVA-like antigens that focus the immune attack. Others are building monoclonal antibodies against HERV-derived NV proteins that tumors aberrantly express, turning the viral relic into a bullseye. Think of it as 2 strategies. Make the tumor shout virus, or paint it with a target the immune system recognizes from ancient training. Meanwhile, combinations multiply. Demethylating drugs with sting agonists to boost the she-gas, sting side of sensing. Checkpoint inhibitors layered with vaccines against ERV peptides. Even adoptive T-cell therapies engineered with receptors that see ERV-derived antigens. The thread through all of it is simple. Use the 8% as a weapon where it hurts the enemy most. The irony lands clean. The same code our cells work so hard to keep quiet can become the flare that calls in help when a cell turns rogue. We're not curing cancer with ghosts. We're forcing cancer to confess in a language the immune system never forgot. Brain wiring and viral switches. From birth onward, the brain's job is timing. What turns on, how long it stays on, and who hears it. Viral switches became part of that clockwork. LTRs that once launched a virus now open in waves in the developing cortex and hippocampus, acting as enhancers and alternative start sites that fine-tune when neurons differentiate, which subtypes expand, and how synapses mature. It's not a single master switch. It's hundreds of local dimmers, coming online just long enough to set thresholds, then going quiet again. Here's what that looks like under the hood. In a progenitor cell, an LTR with a motif for a neuronal pioneer factor shakes off repression, recruits co-activators, and helps loop an enhancer to a promoter through cohesin and CTCF. The target gene's transcription nudges upward at exactly the window that biases a fate decision. More upper layer projection neurons here, a tweak to interneuron programs there. Hours or days later, methylation reasserts, histone marks reset, and the viral switch goes dark, having done its job. Those switches came preloaded with sensitivity. Many LTRs evolved to respond to cellular state, stress, activity, hormones, because viruses needed to sense a cell's machinery. The brain co-opted that responsiveness. When neurons fire, activity-dependent transcription factors bind specific LTRs to modulate genes controlling synapse formation, dendritic spine density, and plasticity molecules. In slices and single-cell data, you see bursts of ERV-derived enhancer activity track with windows of synaptogenesis and critical periods. It's a local chorus that cues the next scene. Then there's the headline act, ARC. This gene is essential for long-term potentiation and memory consolidation. The twist is how it behaves. ARC encodes a protein that oligomerizes into capsid-like shells, structures that resemble retroviral capsids. Those shells package ARC mRNA, bud from one neuron, and fuse with another neuron's membrane to deliver that mRNA. It's a viral delivery trick, repurposed to broadcast, "Here's what I'm changing" across a circuit. Interfering with ARC's capsid formation cripples synaptic plasticity. Letting it run fuels memory. ARC didn't drop in last week. It's a domesticated relic from an ancient retroelement, reshaped to serve neurons. The brain didn't invent a new courier system from scratch. It bent a viral habit to move learning signals between cells. Pair that with ERV-derived enhancers that time when ARC itself is expressed, and you get a feedback loop. Viral-style switches cue plasticity genes, and a retroelement-derived capsid spreads the message. This wiring builds species differences too. Human brains and chimp brains are close cousins, but some LTR families are wired to distinct programs in each lineage. In humans, certain HERV-H elements mark active regulatory hubs in pluripotent stem cells and remain embedded in networks that bias cortical development. Comparative maps show human-specific LTR enhancers near genes involved in neuronal proliferation and migration, potentially nudging cortical expansion and layering. It's not that an ERV made us smart. It's that many small regulatory edits, seeded by viral fossils, steered development down slightly different paths. The same sensitivity that makes these switches useful also makes them fragile. The firewall you heard about matters here. KRAB-ZFPs and TRIM28 put most LTRs under lock and key in mature neurons, keeping viral echoes from muddying precision. But neurons live long. Aging frays methylation fidelity. Inflammatory cytokines from the microglia can leak into neighborhoods where a poised LTR sits. Stress hormones shift transcription factor availability. If an LTR enhancer meant for a developmental window reopens in an adult neuron, it doesn't guide fate. There's no fate left to set. It adds noise. Enough noise, and glia read it as danger. dsRNA from overlapping ERV transcripts, unusual peptides, interferon tone inching up. You can see the system under duress. After traumatic brain injury, some glial populations in human tissue show ERV activation alongside innate immune genes. In mouse models, take away TRIM28 during brain development, and adult brains display ERV-derived proteins and microglial activation. That's not a perfect mirror of human disease, but the mechanism is consistent. Loosen control, viral switches misfire, innate immunity hears virus, and the neighborhood inflames. Flip the perspective, and you find opportunity. Activity-dependent LTRs in defined neuron subtypes become signposts for vulnerability and targets for precision. If a subset of HERV-derived enhancers is consistently open during hippocampal development, variants there could subtly shift memory performance or risk for neurodevelopmental disorders. If an LTR acts as an alternative promoter for a neuronal gene in a subset of interneurons, misregulation could bias excitation-inhibition balance, a theme across psychiatric conditions. This isn't destiny coded by a single fossil. It's a regulatory landscape peppered with viral gear, wired into human programs. Day to day, it's invisible. You learn a song, and synapses strengthen. Behind the scenes, ARC capsid shuttle mRNA, LTRs open and close with neural activity, and KRAB-ZFPs replay their silencing routine to prevent bleed-through. In a life, small differences in how tightly those switches are held, by genetics, by early environment, by aging, shape how resilient circuits are under stress. So when you remember this line tomorrow, credit a quiet alliance. A memory doesn't live in a single cluster of neurons. It lives in changes shared across them, carried by a courier that once belonged to a virus, timed by switches once used to launch an infection. Viral code didn't just visit your brain, it helped wire how your brain changes itself. Evolution's shortcut keys, ERVs across species. While human brains borrowed viral switches and ARC-turned courier, the bigger story is how many species took the same shortcut at different times. Endogenous retroviruses aren't a one-off accident in our lineage. They're a recurring supply drop that evolution keeps raiding. When a retrovirus invades the germline, it doesn't just leave a fossil, it leaves parts, promoters, enhancers, and sometimes a working inverter that other mammals can test drive. Across species, you can watch the same hacks appear independently, pointing to the same pressure and the same payoff. Start with the headline everyone converged on: placenta building. Rodents captured Invid genes multiple times. Syncytin A and Syncytin B in mice are separate domestications, both fusogenic, both essential to their particular placental architecture. Ruminants like cows and sheep landed different in genes again, powering fusion in organs that invade maternal tissue less deeply than primates do, but with huge surface area for exchange. Bats, with flight and high metabolic demands, also independently domesticated ERVs to run their placentas. Carnivores have their versions, different viruses, different epochs, same repurposed engine. The message is blunt. When life needs fusion, it keeps stealing NG. Look past the placenta, and you see waves. In many lineages, bursts of ERV insertions cluster around periods of rapid divergence. They aren't the sole driver of speciation, but they shake the regulatory snow globe. A new set of LTRs arrives with fresh transcription factor motifs, lands near developmental or immune genes, and offers alternative wiring. Over thousands of generations, selection keeps what helps and locks down what harms. The outcome is a chromatin landscape arranged differently in close cousins. Same genes, different dimmers. Immunity showcases that specificity. In primates, an ERV family called MER41 scattered binding sites for STAT1, the interferon-activated switch, near dozens of genes. That seeded interferon-inducible enhancers that now drive AIM2, APOL1, IFI6, SESTM1, and more. Mice don't share MER41. They built similar responsiveness using different elements. The result is species-specific antiviral wiring. Challenge a human macrophage and a mouse macrophage with the same pathogen, and you'll get an overlapping core response with divergences shaped by which retroviral fossils each lineage co-opted. ERVs didn't write the immune system, but they rewrote footnotes in ways that matter in a fight. Reproductive isolation can crystallize from those edits. Imagine two sister species that split 1 million years ago. Each accumulates its own set of ERV insertions and regulatory adoptions. If those changes touch reproductive timing, placental invasion depth, or hormone sensitivity, hybrids might stall. An enhancer wired by an LTR in one species might not respond correctly to transcription factors in the other. A Syncytin-like M captured in one lineage might not find a perfect receptor match in the hybrid placenta, degrading fusion efficiency. None of that requires new genes, just different switches. You can timestamp these events. When the same ERV insertion sits in the same genomic spot across species, orthology, you know the infection predates their split. If the two LTRs flanking a provirus are nearly identical, the insertion is young. If they've drifted far apart, it's older. Recombination between the LTRs often deletes the internal GAG, pol Ms, leaving a solo LTR as a weathered signpost. In humans, HERV-K families retain relatively fresh sequences. A few loci still produce proteins under stress. HERV-H is older, but its LTRs help mark active networks in human pluripotent stem cells, more prominently than in other primates. Those timelines sketch waves of ancient outbreaks layered over evolutionary turns. Parallel to timing is topology. Three-dimensional genome maps show how ERV-derived enhancers don't just boost a neighbor, they can reshape loops. An LTR that recruits CTCF or cooperates with cohesin can flip an entire regulatory neighborhood, bringing a gene into contact with a new enhancer set. Map those contacts in human, chimp, and macaque, and you'll find both conserved scaffolds and species-specific detours. Often, the detours run through retro-element-rich regions. The speed isn't because ERVs are magical, it's because they arrive pre-formatted to plug into transcriptional logic. The immune dance goes both ways. Hosts evolve KRAB-ZFP repertoires that recognize recently active ERV families. That arms race is lineage-specific. A KRAB-ZFP that clamps a young ERV in one primate might be absent in another, changing which families are free to be co-opted as enhancers. Over time, some KRAB-ZFPs themselves become developmentally important, policing not just invaders, but the host programs that now depend on select LTRs. It's a feedback loop. Invader arrives, host evolves a lock, the lock becomes part of normal regulation, new invader requires a new lock. Even behavior leaves fingerprints. Songbirds aren't mammals, but their brains show bursts of transposable element activity associated with vocal learning circuits. In mammals, species with extreme ecologies, naked mole rats underground, bats in flight, carry distinct repeat landscapes and unique ERV domestications aligned to their stress responses and metabolic demands. Those aren't clean cause-effect lines. They're consistent with a model where available viral parts bias, which solutions a lineage can explore quickly. Think of ERVs as shortcut keys on evolution's keyboard. You still need selection, drift, and time. But when a lineage faces a new pressure, diet shift, pathogen, gestation style, verve-derived switches, and the occasional protein give it ready-made modules to test. Most tests fail and get silenced. Enough succeed to leave a trail you can follow from rodents to ruminants to primates. That trail isn't just history. It's a map of what parts biology likes to reuse. Time capsules, and where the next dating ancient outbreaks. To read the fossil record in your DNA, you don't dig, you align. Every ERV insertion is a timestamp if you know where to look. The cleanest signal is orthology. If the same viral scar sits in the exact same spot in human, chimp, and gorilla genomes, that insertion predates our split. If macaques share it too, it's older still. Line up genomes like transparencies, and shared insertions glow as mile markers along the primate family road. But we need clocks, not just markers. Retroviruses arrive with identical, long terminal repeats at both ends, the LTRs that act as promoters. After integration, those twin LTRs start drifting apart, picking up independent mutations at the local rate. Measure their divergence, and you've got a rough sense insertion estimate. Low divergence means young, big divergence means ancient. Sometimes recombination between the two LTRs deletes the interior gag, pol-emd, leaving a lone, solo LTR. That's a weathered gravestone. The infection was old enough for genome housekeeping to tidy up, but the signpost stayed. Now lay families on that timeline. HERV-K elements are among the youngest in humans. Some loci are near intact and under stress, or in certain diseases, they can still produce transcripts and even inbingen. HERV-H sits further back. Few, if any, full proviruses remain, but its LTRs are active players in human pluripotent stem cells, marking regulatory hubs that bias early embryonic programs. The ages aren't trivia. They hint which families had more time to be domesticated as switches, and which remain closer to their viral roots. Orthology adds resolution. Suppose you find an insertion shared by humans and chimps, absent in orangutans. That pins the integration between the orangutan split and the human-chimp split. If an insertion is polymorphic in humans, present in some people, missing in others, you're looking at a very recent event or one still wandering toward fixation. Population genetics picks up the thread. If a young insertion sits at high frequency, it might be hitchhiking with selection. If it lingers at low frequency, drift may be its only ride. Meanwhile, entire waves of insertions line up with ecological turns. In several mammalian lineages, bursts of ERV activity overlap with population bottlenecks or habitat shifts. A small, stressed population with fewer mates and weaker purifying selection can let more insertions survive. Climate swings change pathogen landscapes, opening windows for cross-species jumps. You won't get a day and hour from the genome, but you can map eras when viral traffic surged, then slowed as hosts evolved fresh KRAB-ZFP locks to clamp the newcomers. There's a micro-story hidden in sister species. Picture two close primates splitting on opposite sides of a river that keeps them apart. Each side faces different pathogens and climates. On one bank, a retrovirus finds a way into germ cells and seeds a dozen insertions over a few thousand years. On the other, that virus never crosses. A million years later, their genomes carry different graffiti. Hybridize them in a thought experiment, and you may get mismatched regulation. An enhancer borrowed from an ERV in one lineage won't see the right transcription factors in the other. A KRAB-ZFP repertoire on one side won't recognize the other's younger ERV families. Fertility dips. A regulatory ripple becomes a reproductive barrier. Dating methods stack for confidence. LTR. LTR divergence gives intralocus age. Orthology gives minimum ages across species. Solo LTR frequencies reflect cleanup over time. Even mutation signatures help. Apobec scars are clues that a locus once tried to move or wake and got hammered by host enzymes. Cross-check those signals with phylogenies of the ERV families themselves. Their internal branching patterns mirror past outbreaks, expansions, and die-offs. Tie it back to function. When interferon-responsive MER41 elements landed in the primate lineage, they seeded STAT1 sites near immune genes like AIM2 and APOL1. They aren't in mice. Mice built similar logic using other elements. That pins a regulatory innovation to a primate-specific viral wave. When you see human-biased HERV-H LTRs wiring pluripotency networks, you place that wiring after our split from other apes, shaping early development in a human-leaning way. Timelines aren't just stamps. They're context for why one lineage's cells answer stress, infection, or development cues differently from another's. Medicine borrows these clocks. If a tumor loudly expresses a HERV-K envy, it probably tapped a younger, more intact family. Therapies can target those proteins without hitting most healthy tissues, because older families rarely make proteins. In neurology, evidence of HERV-W in MS lesions fits with a family active in the human line, long enough to scatter copies near glial regulatory neighborhoods, yet old enough that expression is abnormal and inflammatory. Age informs risk. Young families are more likely to wake. Old ones are more likely to be enhancers or landmines if misread. Zoom out once more. Your genome is a layered map of pandemics your ancestors survived. Some insertions are shared souvenirs across primates. Others are human-only scars. Their positions and ages chronicle waves of invasion, adjustment, and truce. Those dates don't sit in textbooks. They sit in your cells, telling you which ghosts are likely to sing and which already joined the choir. With the map drawn, the next move is obvious. Test the volume knobs. Scientists, with volume knobs, CRISPR, and epigenetic editing. Now that we've mapped which ghosts sing and when, the question becomes, can we ride the faders without wrecking the soundboard? That's the promise and the risk. We don't delete the 8%. We tune it, precisely, briefly, and in the right rooms. Start with CRISPR, but not the cut and paste kind. The workhorse here is a deactivated Cas9, dCas9, that still finds a DNA sequence but doesn't slice it. Bolt on a silencer domain like KRAB, and you get CRISPR interference. CRISPR. Guide RNA steer dCas9 CRAB to a specific LTR or ERV promoter. Park it there, and KRAB recruits the same repression machinery cells already trust. Trim 28s, CAP1, SETVB1, H3K9, Me3 writers. The locus goes dim without changing the letters. Turn off the guide, and expression can rebound. It's a reversible mute button. Flip the polarity, and you get the opposite tool. dCas9 fused to activators, VP64, P300, VPR, can wake a chosen LTR in a chosen cell type. Not to cause chaos, but to test causality. Does opening this interferon-responsive MER41 element next to AIM2 amplify the alarm? Does damping it cool an overzealous macrophage? With CRISPR and CRISPR, you don't argue in abstracts. You run the program and watch the readouts. Single-cell RNA-seq to see which genes move, ATAC-seq to check chromatin opening, ChIP-seq to confirm the right marks land where you aimed. Base editors add nuance. They don't break DNA, they swap letters, C to T, A to G, within a tight window. If an LTR's key transcription factor motif is the problem, a single base tweak can defang it without touching nearby genes. No double-strand breaks, fewer rearrangements, smaller blast radius. Where methylation is the main lock, tethering TET1 to DKase9 can demethylate a promoter on purpose, fusing DNMT3. The buts therefore chain, trade-offs, we can't escape. Here's the dilemma, in plain terms. We mute ERVs to cool autoimmune fire, but that same muting may dim the viral siren we use to expose tumors. Therefore, control can't be global. It has to be specific, time, tissue, and context. The genome doesn't offer a single master switch. It hands us a panel of sliders, each tied to a neighborhood, a cell state, and a risk profile. Start with autoimmunity and neuroinflammation. If HERV-W NV fuels microglial activation in MS lesions, neutralizing NV with an antibody could reduce damage. But if we extend that strategy broadly, lowering interferon tone system-wide, we might also blunt antiviral defenses and shrink the very window where viral mimicry helps immunotherapy. Therefore, treatment should be staged. Aim at NV locally or temporally in MS flares, while preserving innate sensing elsewhere, especially in patients who may also need tumor surveillance. Cancer flips the sign. Demethylating a tumor to wake ARV dsRNA makes it visible. But methylation drugs don't read intentions. They act where delivered. Therefore, dose low, pulse briefly, and confine exposure to the tumor microenvironment when possible. Pair with checkpoint inhibitors during the interferon on window, then back off before collateral inflammation exhausts T cells. The same ERV signal that flags cancer can, if sustained, breed resistance. Pregnancy adds non-negotiable constraints. Syncytin 1 and Syncytin 2 must sing in trophoblasts and stay silent elsewhere. Immune checkpoint therapy can endanger that balance. Demethylating drugs can too. Therefore, reproductive status and intent aren't side notes, they're gating criteria. Screen, counsel, and, where needed, postpone ERV modulating treatments until the placenta's program isn't at risk. Aging complicates everything. Methylation fidelity phrase, KRAB-ZFP policing varies person to person. Therefore, a 70-year-old with chronic inflammation may need lighter touches than a 30-year-old, even with the same diagnosis. Clinical protocols should incorporate methylation age and interferon tone, not just birthdays and lab ranges. Personalization starts with an ERV fingerprint, locus-level expression, family patterns, and peptide presentation. Layer on interferon markers, methylation maps, and single-cell readouts that separate cancer cells from stroma and neurons from glia. Therefore, dosing isn't a guess. It's informed by whether MER41-linked enhancers are already hot, whether HERV-K peptides sit on MHC, and whether dsRNA burden is high enough to merit a push or already near the ceiling. There's also this action window. Vaccines and acute infections transiently spike interferon. That's good for defense, and in some tissues, flips ERV-seeded enhancers on purpose. But the same wave could aggravate a smoldering autoimmune niche. Therefore, plan around peaks. Schedule ERV-targeting antibodies or demethylating pulses away from vaccine days. Monitor symptoms in the weeks after respiratory infections. Proactively adjust steroids or anti-cytokine meds in high-risk patients. In neurology, precision matters down to cell types. Neurons can't tolerate chronic interferon. Microglia can mount it, but too much prunes synapses. Therefore, delivery should favor microglia when dampening HERV-W signaling and avoid broad agents that cross the blood-brain barrier indiscriminately if they risk silencing activity-dependent LTRs that tune plasticity. A targeted antibody against NV is cleaner than a global epigenetic suppressant in the brain. Ethics rides alongside engineering. CRISPR at an LTR during a flare is one thing. Germline edits to excise ERVs are another. Those sequences also scaffold development and immunity. Therefore, edits must be somatic, reversible where possible, and monitored with assays that catch off target rewiring, ATAC-seq for chromatin shifts, CHIP-seq for misplaced marks, and single-cell RNA-seq for ectopic programs. Now the cross-pressure that will define real clinics. A patient with melanoma responds to checkpoint therapy, partly because her tumor screams ERV. Months later, she develops autoimmune thyroiditis. If we hammer interferon to cool the autoimmunity, we might kill the signal that keeps T cells on task against the cancer. Therefore, we titrate local steroids, temporary PD-1 pause, narrow-spectrum cytokine blockers, while maintaining the ERV-driven visibility that correlates with her tumor control. It's not elegant, it's necessary. Biomarkers make these calls less blind. An ERV activity score, an interferon signature, methylation age, and circulating NV protein levels become a dashboard. Therefore, we can preset guardrails. If ENV rises without matched dsRNA alarms, think antibody. If dsRNA and MDA5 signaling dominate without NFRU protein, think demethylating pulses plus checkpoint. If both rise in a pregnant patient, stop. Risk outweighs gain. Monitoring windows matter. Pregnancy and postpartum, adolescence's cortical refinement, midlife immune drift, and late life inflammaging are periods when ERV control naturally shifts. Therefore, any intervention that touches ERV expression should respect these calendars with lower doses, tighter follow-up, and rapid off-ramps. Finally, be honest about trade-offs. We can't code away. Silence too much, and you blunt adaptability. Tumor surveillance, antiviral spikes, plasticity tuning. Wake too much, and you court autoimmunity, neuroinflammation, and fusion in the wrong tissue. Therefore, our job isn't to choose silence or noise. It's to learn the music. Who needs to sing, when, and for how long, and ride the faders so the performance holds. Your body, today. Practical meaning without hype. Let's bring this home. You're carrying around an archive that can help or hurt depending on how tightly it's locked. You don't control the code, but you do influence the locks. Think levers, not miracles. Start with inflammation. Chronic, low-grade inflammation tugs at epigenetic marks that keep many ERV loci quiet. Cytokines like interferons were built to flip specific LTRs in immune cells on purpose. Sprinkle those same signals for months and repression phrase in places it shouldn't. The most practical move is to shrink the baseline inflammation you carry. That's sleep, stress management, movement, avoiding smoke, and managing infections early. Boring advice with molecular backing. Sleep first. During consolidated sleep, hormones and autonomic tone shift toward repair. Studies mapping methylation patterns show that regular, sufficient sleep predicts tighter control at repeat elements, including LTRs. Miss nights chronically, and methylation variability creeps up. In brain tissue, that variability means more noise near regulatory elements that should stay quiet after development. You won't feel methyl groups moving, but you'll feel brain fog, immune weirdness, or a shorter fuse to stress. All conditions that correlate with loosened control. Stress next. Acute stress is part of life. Chronic stress is a chemistry bath. Glucocorticoids change transcription factor availability and nudge chromatin remodelers. In immune cells, that can distort interferon programs and push miR41-linked enhancers harder or longer than designed. Real tactics beat platitudes. Carve out a repeatable downshift ritual. Breathing, a walk, a call, so stress spikes.